Introduction to human reproduction. Human reproduction is an intricate biological process fundamental to the continuation of our species. It encompasses a series of complex events that span from the formation of gametes to the development of a new individual. This process is not only essential for perpetuating life, but also for maintaining genetic diversity, which is crucial for the adaptation and survival of the human species. At its core, reproduction involves two primary phases: the formation of gametes and the subsequent fertilization of these gametes to produce a zygote. Gametes are specialized sex cells, sperm in males and eggs, or ova, in females, that carry half of the genetic information required to create a new individual. The fusion of these gametes during fertilization restores the full complement of chromosomes, initiating the development of a new organism. The formation of gametes, a process known as gametogenesis, begins with meiosis, a type of cell division that reduces the chromosome number by half. In males, this process takes place in the testes, resulting in the production of sperm cells. In females, it occurs in the ovaries, producing eggs. Each gamete carries a unique combination of genes due to the random assortment of chromosomes and genetic recombination that occurs during meiosis. This genetic variation is a key factor in the diversity of traits among offspring and contributes to the evolutionary adaptability of the species. Once gametes are produced, they must come together in the process of fertilization. Fertilization typically occurs in the fallopian tubes, where a sperm cell meets and penetrates an egg. This union forms a zygote, a single cell with a complete set of chromosomes, half from the mother and half from the father. The zygote then begins a series of mitotic divisions, developing into a blastocyst that implants into the uterine lining. This implantation marks the beginning of pregnancy and the transition from a single-cell zygote to a complex multicellular embryo. Embryonic development follows, characterized by the formation of three primary germ layers: the ectoderm, mesoderm, and endoderm. These layers give rise to all the tissues and organs of the body. The embryonic period is crucial as it sets the foundation for the formation of major organs and body systems. Following embryonic development, the fetal stage begins, during which the organs mature and grow. This period is marked by significant growth and refinement of body structures, preparing the fetus for survival outside the womb. Reproductive processes are regulated by a complex interplay of hormones. In males, testosterone plays a central role in regulating sperm production and maintaining secondary sexual characteristics. In females, hormones such as estrogen and progesterone regulate the menstrual cycle, ovulation, and pregnancy. Gonadotropins, FSH and LH, from the pituitary gland also play critical roles in controlling reproductive functions. The birth process, or parturition, involves several stages: labor, delivery, and the transition to neonatal life. Labor begins with contractions of the uterine muscles, leading to the dilation of the cervix. Delivery follows as the baby moves through the birth canal. The transition from fetal to neonatal circulatory systems is a remarkable physiological adjustment, necessary for the newborn's independent survival. Postnatal development involves continued growth and maturation. After birth, infants undergo rapid physical and cognitive development. Motor skills, sensory perception, and cognitive abilities evolve as the newborn adapts to life outside the womb. This period of development is critical for laying the groundwork for future learning and health. Understanding human reproduction and development is essential for appreciating how life begins and evolves. It provides insights into the biological processes that ensure the continuation of life and highlights the intricate systems that support human growth from conception through maturity. In the subsequent sections, we will delve deeper into each aspect of this fascinating process, exploring the anatomical, physiological, and developmental details that shape human life. Reproductive anatomy, male and female systems. The human reproductive system is a complex network of organs and structures designed to facilitate the production, transport, and union of gametes, ultimately leading to reproduction. Understanding the anatomy of the male and female reproductive systems is crucial for grasping the broader concepts of human reproduction and development. Male reproductive system. The male reproductive system is primarily concerned with the production and delivery of sperm, the male gamete. Key structures in this system include testes. The testes are oval-shaped organs located in the scrotum, a pouch of skin hanging outside the body. They are responsible for producing sperm through a process called spermatogenesis. The testes also produce testosterone, a hormone crucial for the development of male secondary sexual characteristics and the regulation of sperm production. Epididymis. Each testis is connected to an epididymis, a coiled tube where sperm mature and are stored. The epididymis is essential for sperm to gain motility and the ability to fertilize an egg. Vas deferens. The vas deferens is a muscular tube that transports sperm from the epididymis to the ejaculatory duct. It is part of the pathway through which sperm travel during ejaculation. Ejaculatory duct. The ejaculatory duct is formed by the union of the vas deferens and the seminal vesicle duct. It passes through the prostate and empties into the urethra. This duct is where sperm mixes with seminal fluid to form semen. Urethra. The urethra is a tube that runs through the penis and serves as the conduit for both urine and semen. During ejaculation, sperm and seminal fluid are expelled through the urethra. Accessory glands. Seminal vesicles. These glands secrete a significant portion of the seminal fluid, which provides nutrients and energy for sperm. Prostate gland. The prostate gland produces a fluid that helps to nourish and transport sperm and also helps to neutralize the acidic environment of the female reproductive tract. Bulbourethral glands. Cooper's glands. These glands secrete a pre-ejaculatory fluid that lubricates the urethra and neutralizes any residual acidity. Female reproductive system. The female reproductive system is designed for the production of eggs, fertilization, and nurturing of the embryo. Key structures include ovaries. The ovaries are almond-shaped organs located in the pelvic cavity. They are responsible for producing eggs, ova, and releasing them during the menstrual cycle. The ovaries also produce hormones such as estrogen and progesterone, which regulate the menstrual cycle and support pregnancy. Fallopian tubes, also known as uterine tubes, these are narrow tubes that extend from the upper corners of the uterus to the ovaries. The fallopian tubes are the site of fertilization, where sperm meets the egg. They transport the fertilized egg to the uterus for implantation. Uterus. The uterus is a pear-shaped, muscular organ located in the pelvic cavity. It has three main parts: the fundus, upper part, the body, main part, and the cervix, lower part. The uterus is where the fertilized egg implants and develops into a fetus during pregnancy. It also plays a crucial role during childbirth, contracting to help deliver the baby. Cervix. The cervix is the lower, narrow part of the uterus that connects to the vagina. It serves as a passageway for sperm to enter the uterus and for menstrual blood to exit. During childbirth, the cervix dilates to allow the baby to pass through the birth canal. Vagina. The vagina is a muscular, elastic tube that extends from the cervix to the external genitalia. It serves as the passageway for menstrual flow, sexual intercourse, and childbirth. External genitalia. The external genitalia, or vulva, includes the labia majora, labia minora, clitoris, and vaginal opening. These structures protect the internal reproductive organs and provide the site for sexual intercourse. Understanding the anatomy of the male and female reproductive systems provides a foundation for comprehending how gametes are produced, transported, and how fertilization occurs. It also highlights the complexity of the processes involved in human reproduction and development, setting the stage for further exploration of these fascinating biological systems. Gametogenesis, formation of gametes. Gametogenesis is the process by which specialized cells known as gametes are produced. In humans, this process occurs in the testes for males and the ovaries for females, leading to the formation of sperm and eggs, respectively. Gametogenesis is crucial for sexual reproduction as it ensures genetic diversity and the continuation of the species. Spermatogenesis, formation of sperm. Spermatogenesis is the process by which sperm cells are produced in the testes. This complex process involves several stages. Formation of spermatogonia. Spermatogenesis begins with spermatogonia, which are diploid stem cells located in the seminiferous tubules of the testes. These cells undergo mitotic divisions to produce more spermatogonia, ensuring a continuous supply of cells for sperm production. Meiosis I. Spermatogonia differentiate into primary spermatocytes, which are diploid cells. Each primary spermatocyte undergoes meiosis I, resulting in two haploid secondary spermatocytes. Meiosis I reduces the chromosome number by half, creating cells with only one set of chromosomes. Meiosis II. Each secondary spermatocyte then undergoes meiosis II, producing two haploid spermatids per secondary spermatocyte. This means that each primary spermatocyte ultimately yields 4 haploid spermatids. Spermiogenesis. Spermatids undergo a transformation process called spermiogenesis, during which they mature into spermatozoa, sperm cells. This process involves the development of a flagellum, tail, for motility, condensation of the nucleus, and shedding of excess cytoplasm. Mature sperm cells are streamlined and designed for efficient delivery of genetic material to the egg. Sperm transport and storage. Once mature, sperm are transported to the epididymis, where they are stored and undergo further maturation. During ejaculation, sperm travel through the vas deferens, ejaculatory duct, and urethra to be released. Oogenesis, formation of eggs. Oogenesis is the process by which eggs, ova, are produced in the ovaries. Unlike spermatogenesis, oogenesis begins before birth and involves a series of complex stages. Formation of oogonia. During fetal development, oogonia, diploid stem cells, are formed in the ovaries. These cells undergo mitotic divisions to increase their numbers. Primary oocytes. Oogonia differentiate into primary oocytes, which enter meiosis I and become arrested in prophase I of meiosis. This arrest occurs during fetal development and continues until puberty. Meiosis resumption. At puberty, during each menstrual cycle, a primary oocyte resumes meiosis I. It completes meiosis I to produce a secondary oocyte and a smaller polar body. The polar body is a non-functional cell that eventually degenerates. Secondary oocyte. The secondary oocyte begins meiosis II but is arrested in metaphase II. It is released from the ovary during ovulation. If fertilization occurs, meiosis II completes, resulting in a mature ovum and a second polar body. Completion of meiosis. If the secondary oocyte is fertilized by sperm, it completes meiosis II, producing an ovum with a full set of chromosomes and a second polar body. The fertilized ovum, zygote, then begins the process of early embryonic development. Importance of gametogenesis. Gametogenesis is essential for sexual reproduction as it ensures the production of gametes with half the chromosome number of somatic cells. This reduction in chromosome number is achieved through meiosis, a specialized form of cell division. By creating haploid cells, gametogenesis enables the combination of genetic material from two parents, leading to genetic diversity in offspring. Additionally, gametogenesis involves mechanisms of genetic recombination and independent assortment, which further contribute to genetic variation. This variation is vital for the adaptability and evolution of populations. In summary, Gametogenesis is a fundamental biological process that results in the formation of sperm and eggs, setting the stage for fertilization and the continuation of human life. The detailed processes of spermatogenesis and oogenesis highlight the complexity and precision of reproductive biology, ensuring the successful creation of new individuals and the perpetuation of genetic material. Fertilization and early development. Fertilization marks the critical moment when a sperm cell successfully merges with an egg cell, resulting in the formation of a zygote. This process initiates the complex journey of early human development, leading to the formation of a new individual. The stages of fertilization and early development are fundamental to understanding how a single cell transforms into a multicellular organism. Fertilization. Fertilization typically occurs in the fallopian tubes, which serve as the conduit between the ovary and the uterus. The process can be divided into several key steps. Sperm capacitation. Before fertilization, sperm undergo a series of biochemical changes known as capacitation. This process, which occurs in the female reproductive tract, involves alterations to the sperm's membrane that enhance its ability to penetrate the egg's outer layers. Sperm egg recognition. The sperm must navigate through the female reproductive tract to reach the egg. Once at the egg, sperm encounter the zona pellucida, a glycoprotein layer surrounding the egg. Specific proteins on the sperm's surface interact with receptors on the zona pellucida, allowing the sperm to bind and initiate the acrosome reaction. Acrosome reaction. During this reaction, enzymes are released from the acrosome, a cap-like structure on the sperm's head, that digests the zona pellucida, enabling the sperm to penetrate the egg. Once a sperm successfully breaches the zona pellucida, it merges with the egg's plasma membrane. Fusion of gametes. Upon successful entry, the sperm's nucleus enters the egg, and its genetic material combines with that of the egg. This fusion forms a single diploid cell known as a zygote, which contains a complete set of chromosomes, half from the mother and half from the father. Prevention of polyspermy. To prevent multiple sperm from fertilizing the egg undergoes a series of changes that block additional sperm entry. This process is essential to maintain the correct number of chromosomes in the resulting zygote. Early development. Following fertilization, the zygote undergoes a series of developmental stages as it travels down the fallopian tube towards the uterus. Cleavage. The zygote begins a series of rapid mitotic divisions known as cleavage. These divisions do not increase the overall size of the embryo but instead produce smaller cells called blastomeres. This process results in the formation of a solid ball of cells known as a morula. Blastocyst formation. As cleavage continues, the morula transforms into a blastocyst. The blastocyst is a hollow sphere with an inner cell mass, which will develop into the embryo, and an outer layer called the trophoblast, which will form part of the placenta. Implantation. The blastocyst reaches the uterus and adheres to the uterine lining, a process known as implantation. The trophoblast cells invade the uterine lining, establishing a connection between the developing embryo and the mother's blood supply. Successful implantation is crucial for the continuation of pregnancy. Gastrulation. Following implantation, the inner cell mass of the blastocyst undergoes gastrulation, a process in which it differentiates into three primary germ layers: the ectoderm, mesoderm, and endoderm. These germ layers give rise to all of the body's tissues and organs. Formation of the embryo. As the embryo continues to develop, it begins to form distinct structures and organs. This period is marked by significant morphological changes and the establishment of the basic body plan. Importance of early development. The stages of fertilization and early development are critical for establishing the groundwork for a healthy pregnancy and the formation of a viable individual. Any disruptions in these early stages can lead to developmental abnormalities or pregnancy loss. Understanding these processes provides insight into the intricate mechanisms that drive human development from a single cell to a fully formed embryo ready for further growth. In summary, fertilization and early development are foundational to human reproduction. The successful fusion of sperm and egg initiates a series of events that lead to the formation and implantation of a blastocyst. This early development is pivotal in setting the stage for further embryonic growth and the eventual emergence of a new individual. Embryonic and fetal development. Human development is a remarkable journey that begins with fertilization and progresses through distinct stages, culminating in the birth of a new individual. This process is characterized by two major phases: embryonic development and fetal development. Each stage involves complex processes that transform a single-cell zygote into a fully formed fetus ready for birth. Embryonic development. Embryonic development encompasses the first 8 weeks of gestation, a period marked by rapid and fundamental changes. During this stage, the embryo undergoes critical processes that establish the basic body plan and organ systems. Germ layer formation. The early stages of embryonic development involve the formation of 3 primary germ layers through a process called gastrulation. Ectoderm. This outer layer gives rise to the nervous system, brain and spinal cord, skin and hair. Mesoderm. The middle layer forms muscles, bones, the cardiovascular system, heart and blood vessels, and other connective tissues. Endoderm. The innermost layer develops into the gastrointestinal tract, respiratory system, and other internal organs. Organogenesis. Following the establishment of germ layers, organogenesis begins. This process involves the development and differentiation of organs and tissues. Major organs such as the heart, liver, and kidneys start to form, and the embryo's basic body plan becomes recognizable. Formation of the primitive streak. The primitive streak appears as a linear structure on the embryonic surface and plays a crucial role in establishing the embryo's bilateral symmetry and the anterior-posterior axis. It marks the beginning of the embryonic development of body structure and pattern. Development of major systems. By the end of the embryonic period, the embryo has developed rudimentary forms of major systems, including the cardiovascular system, digestive system, and central nervous system. Limb buds appear, and facial features become more distinct. Embryo to fetus transition. At around 8 weeks, the developing organism is referred to as a fetus. This transition signifies the end of the embryonic period and the beginning of the fetal stage, characterized by further growth and maturation of the organ systems established during embryonic development. Fetal development. Fetal development spans from the 9th week of gestation until birth. This stage is marked by significant growth and refinement of the organs and systems that were formed during the embryonic period. Growth and maturation. During the fetal stage, the fetus undergoes substantial growth. The organs and systems that began to form in the embryonic stage continue to develop and mature. The heart becomes fully functional, the lungs develop further, and the digestive system prepares for independent function after birth. Sensory and motor development. As the fetus grows, it begins to develop sensory and motor functions. By around 16 weeks, the fetus can move its limbs and respond to external stimuli. By the end of the 2nd trimester, the fetus can detect light and sounds, and its movements become more coordinated. Preparation for birth. In the final trimester, the fetus continues to grow in size and gain weight. The lungs mature further, producing surfactant to facilitate breathing air at birth. The fetus also undergoes changes in body fat distribution, which helps regulate body temperature after birth. Fetal monitoring. Throughout fetal development, various techniques are used to monitor the health and development of the fetus. Ultrasound imaging provides detailed views of fetal growth and anatomy, while other tests assess fetal heart rate and overall well-being. Birth readiness. As the fetus reaches full term, It prepares for birth. The position of the fetus shifts into the optimal position for delivery, and the body undergoes final adjustments to transition from the fetal environment to the external world. Importance of embryonic and fetal development. The processes of embryonic and fetal development are crucial for ensuring the proper formation and functioning of the organs and systems necessary for life outside the womb. Disruptions or abnormalities during these stages can lead to developmental disorders or pregnancy complications. Understanding embryonic and fetal development provides insights into the complex processes that shape human growth and highlights the critical periods when interventions can positively impact prenatal health. This knowledge is essential for advancing prenatal care and supporting healthy pregnancies. In summary, embryonic and fetal development are fundamental stages in human growth, marked by intricate processes that transform a zygote into a fully developed fetus. Each stage involves critical developments that establish the foundation for a healthy birth and future life. Embryonic and fetal development. Human development is a remarkable journey that begins with fertilization and progresses through distinct stages, culminating in the birth of a new individual. This process is characterized by two major phases: embryonic development and fetal development. Each stage involves complex processes that transform a single-cell zygote into a fully formed fetus ready for birth. Embryonic development. Embryonic development encompasses the first 8 weeks of gestation, a period marked by rapid and fundamental changes. During this stage, the embryo undergoes critical processes that establish the basic body plan and organ systems. Germ layer formation. The early stages of embryonic development involve the formation of 3 primary germ layers through a process called gastrulation. Ectoderm. This outer layer gives rise to the nervous system, brain and spinal cord, skin and hair. Mesoderm. The middle layer forms muscles, bones, the cardiovascular system, heart and blood vessels, and other connective tissues. Endoderm. The innermost layer develops into the gastrointestinal tract, respiratory system, and other internal organs. Organogenesis. Following the establishment of germ layers, organogenesis begins. This process involves the development and differentiation of organs and tissues. Major organs such as the heart, liver, and kidneys start to form, and the embryo's basic body plan becomes recognizable. Formation of the primitive streak. The primitive streak appears as a linear structure on the embryonic surface and plays a crucial role in establishing the embryo's bilateral symmetry and the anterior-posterior axis. It marks the beginning of the embryonic development of body structure and pattern. Development of major systems. By the end of the embryonic period, the embryo has developed rudimentary forms of major systems, including the cardiovascular system, digestive system, and central nervous system. Limb buds appear, and facial features become more distinct. Embryo to fetus transition. At around 8 weeks, the developing organism is referred to as a fetus. This transition signifies the end of the embryonic period and the beginning of the fetal stage, characterized by further growth and maturation of the organ systems established during embryonic development. Fetal development. Fetal development spans from the 9th week of gestation until birth. This stage is marked by significant growth and refinement of the organs and systems that were formed during the embryonic period. Growth and maturation. During the fetal stage, the fetus undergoes substantial growth. The organs and systems that began to form in the embryonic stage continue to develop and mature. The heart becomes fully functional, the lungs develop further, and the digestive system prepares for independent function after birth. Sensory and motor development. As the fetus grows, it begins to develop sensory and motor functions. By around 16 weeks, the fetus can move its limbs and respond to external stimuli. By the end of the 2nd trimester, the fetus can detect light and sounds, and its movements become more coordinated. Preparation for birth. In the final trimester, the fetus continues to grow in size and gain weight. The lungs mature further, producing surfactant to facilitate breathing air at birth. The fetus also undergoes changes in body fat distribution, which helps regulate body temperature after birth. Fetal monitoring. Throughout fetal development, various techniques are used to monitor the health and development of the fetus. Ultrasound imaging provides detailed views of fetal growth and anatomy, while other tests assess fetal heart rate and overall well-being. Birth readiness. As the fetus reaches full term, it prepares for birth. The position of the fetus shifts into the optimal position for delivery, and the body undergoes final adjustments to transition from the fetal environment to the external world. Importance of embryonic and fetal development. The processes of embryonic and fetal development are crucial for ensuring the proper formation and functioning of the organs and systems necessary for life outside the womb. Disruptions or abnormalities during these stages can lead to developmental disorders or pregnancy complications. Understanding embryonic and fetal development provides insights into the complex processes that shape human growth and highlights the critical periods when interventions can positively impact prenatal health. This knowledge is essential for advancing prenatal care and supporting healthy pregnancies. In summary, embryonic and fetal development are fundamental stages in human growth, marked by intricate processes that transform a zygote into a fully developed fetus. Each stage involves critical developments that establish the foundation for a healthy birth and future life. Hormonal regulation of reproduction. Hormonal regulation is fundamental to human reproduction, orchestrating the complex processes of gametogenesis, fertilization, pregnancy, and childbirth. Hormones are chemical messengers produced by endocrine glands that travel through the bloodstream to target organs, influencing various physiological functions. In reproduction, hormones regulate the development and function of the reproductive systems, coordinate the menstrual cycle, and support pregnancy. Hormones in the male reproductive system. Testosterone. Testosterone is the primary male sex hormone produced by the testes. It plays a crucial role in the development of secondary sexual characteristics during puberty, such as facial hair and a deeper voice. Testosterone is also essential for spermatogenesis, the process of sperm production. It acts on the Sertoli cells in the seminiferous tubules to support sperm maturation and influence libido and erectile function. Luteinizing hormone, LH, and follicle-stimulating hormone, FSH. LH and FSH are produced by the anterior pituitary gland and regulate testosterone production and spermatogenesis. LH stimulates the Leydig cells in the testes to produce testosterone, while FSH, along with testosterone, supports the Sertoli cells in the seminiferous tubules, promoting sperm production. Inhibin. Inhibin is a hormone produced by the Sertoli cells in response to FSH. It inhibits the production of FSH by the pituitary gland through negative feedback, helping to regulate sperm production and maintain hormonal balance. Hormones in the female reproductive system. Estrogen. Estrogen is primarily produced by the ovaries and is crucial for the development of secondary sexual characteristics in females, such as breast development and the regulation of the menstrual cycle. It stimulates the growth and maturation of ovarian follicles and plays a key role in the proliferation and maintenance of the endometrial lining of the uterus, preparing it for potential implantation of a fertilized egg. Progesterone. Progesterone is produced by the corpus luteum in the ovary after ovulation. It prepares the endometrium for implantation and supports early pregnancy by maintaining the uterine lining. If pregnancy does not occur, progesterone levels fall, leading to the shedding of the endometrial lining and the onset of menstruation. Luteinizing hormone, LH, and follicle-stimulating hormone, FSH. LH and FSH are produced by the anterior pituitary gland and regulate the female reproductive cycle. FSH stimulates the growth of ovarian follicles, while LH triggers ovulation, the release of a mature egg from the ovary. The interplay between LH and FSH orchestrates the menstrual cycle and the release of hormones from the ovaries. Human chorionic gonadotropin, HCG, during pregnancy. The developing placenta produces HCG, which is detected in pregnancy tests. HCG maintains the corpus luteum, ensuring continued production of progesterone and estrogen during the early stages of pregnancy. This support is essential for maintaining the uterine lining and promoting fetal development. Hormonal regulation of the menstrual cycle. The menstrual cycle, a monthly process of ovulation and menstruation, is regulated by a complex interaction of hormones. Follicular phase. Beginning on the first day of menstruation, FSH stimulates the growth of ovarian follicles. Estrogen levels rise as the follicles mature, leading to the thickening of the endometrial lining. Ovulation. A surge in LH triggers ovulation, the release of a mature egg from the ovary. The increase in estrogen levels just before the surge also plays a role in signaling ovulation. Luteal phase. Following ovulation, the corpus luteum forms and produces progesterone. This hormone maintains the endometrial lining for potential implantation. If fertilization does not occur, the corpus luteum degenerates, leading to a drop in progesterone and estrogen levels, causing menstruation. Hormonal regulation during pregnancy and birth. Pregnancy. Early in pregnancy, rising levels of estrogen and progesterone from the placenta support the uterine lining and fetal development. Hormones also regulate various physiological changes, such as increased blood volume and metabolic adjustments. Birth. Hormonal changes in late pregnancy prepare the body for labor. Oxytocin, produced by the pituitary gland, stimulates uterine contractions and plays a role in the initiation of labor. The release of prostaglandins also contributes to the cervical ripening and contraction process. In summary, hormonal regulation of reproduction involves a sophisticated network of endocrine signals that manage gametogenesis, the menstrual cycle, pregnancy, and childbirth. Hormones like testosterone, estrogen, progesterone, LH, FSH, and HCG play crucial roles in maintaining reproductive health and ensuring successful reproduction. Understanding these hormonal processes provides insight into the intricate mechanisms that govern human reproduction. Birth and postnatal development. The transition from fetal life to the external world is a complex process involving the birth of the baby and the subsequent stages of postnatal development. This period is characterized by significant physiological and developmental changes that support the newborn's adaptation to life outside the womb and lay the foundation for future growth and development. Birth process. The birth process, also known as labor and delivery, involves several stages that lead to the delivery of the baby. These stages include labor. Labor is divided into 3 stages. First stage. This stage begins with the onset of regular contractions and ends when the cervix is fully dilated to 10 centimeters. It involves 2 phases. The latent phase, where contractions are relatively mild and infrequent, and the active phase, where contractions become stronger and more frequent. During this stage, the cervix gradually effaces, thins, and dilates, allowing the baby to move into the birth canal. Second stage. The second stage of labor begins when the cervix is fully dilated and ends with the delivery of the baby. This stage is characterized by intense contractions that help push the baby through the birth canal. The mother may be instructed to push with each contraction to facilitate the baby's descent. As the baby's head emerges, it rotates to navigate the birth canal and is eventually delivered. Third stage. The third stage of labor involves the delivery of the placenta. After the baby is born, contractions continue to help detach and expel the placenta from the uterine wall. This stage is crucial for ensuring that no placental tissue remains in the uterus, which could lead to complications. The neonatal transition. Following birth, the newborn undergoes several physiological adjustments to adapt to life outside the womb. This includes initiating breathing, adjusting circulation, and regulating body temperature. The newborn's lungs fill with air, and the circulatory system changes from fetal to postnatal circulation, with the closure of fetal shunts such as the ductus arteriosus and foramen ovale. APGAR score. Immediately after birth, the APGAR score is used to assess the newborn's overall health. This score evaluates 5 criteria: heart rate, respiratory effort, muscle tone, reflex response, and skin color. Each criterion is scored from 0 to 2, with a maximum total score of 10. The APGAR score helps determine if any immediate medical intervention is needed. Postnatal development. Postnatal development encompasses the growth and maturation of the newborn during the first year of life. This period is characterized by rapid physical, motor, and cognitive development. Physical growth. Newborns experience rapid weight gain and growth during the first year. Infants typically double their birth weight by around 5 to 6 months and triple it by their first birthday. They also grow in length, with an average increase of about 10 inches during the first year. Regular pediatric checkups monitor growth patterns and ensure healthy development. Motor development. Motor skills develop sequentially from reflexive movements to voluntary actions. In the first few months, infants exhibit reflexes such as the grasp reflex and rooting reflex. By around 4 to 6 months, they begin to reach for objects, roll over, and sit with support. By 9 to 12 months, most infants can crawl, stand with assistance, and take their first steps. Cognitive and sensory development. Cognitive development involves the acquisition of skills such as attention, memory, and problem-solving. Newborns begin to recognize familiar faces and voices and respond to sensory stimuli. By 6 to 12 months, they develop object permanence, the understanding that objects continue to exist even when out of sight. They also begin to engage in simple problem-solving and social interactions. Social and emotional development. Social and emotional development is marked by the establishment of early relationships and the expression of emotions. Infants form strong attachments to primary caregivers and display a range of emotions, including joy, anger, and fear. Social interactions become more complex as infants begin to engage in reciprocal play and show preference for familiar people. Nutrition and feeding. Proper nutrition is essential for healthy postnatal development. Breastfeeding provides optimal nutrition and immunological protection for newborns. As infants grow, they transition to complementary foods, which support continued growth and development. Conclusion. The birth process and postnatal development are critical phases in human life, involving significant physiological, motor, cognitive, and emotional changes. Successful transition from fetal life to the external environment and the subsequent growth and development of the newborn lay the foundation for a healthy and thriving individual. Understanding these processes highlights the importance of prenatal care, the birth experience, and early postnatal support in promoting optimal development and well-being. Key concepts of the chapter. The study of human reproduction and development involves a deep understanding of various fundamental concepts that underpin the processes of creating and nurturing a new individual. This section will summarize the key concepts of reproductive anatomy, developmental stages, and the intricate hormonal regulations that guide these processes. Reproductive anatomy. Male reproductive system. The male reproductive system is designed for the production and delivery of sperm. It includes testes, located in the scrotum. The testes produce sperm and hormones like testosterone. Epididymis, a coiled tube where sperm mature and are stored. Vas deferens transports sperm from the epididymis to the ejaculatory duct. Accessory glands, including the seminal vesicles, prostate gland, and bulbourethral glands, which produce fluids that nourish and transport sperm. The male reproductive system operates under the regulation of hormones such as testosterone, luteinizing hormone, LH, and follicle-stimulating hormone, FSH, which collectively influence sperm production and secondary sexual characteristics. Female reproductive system. The female reproductive system is structured to produce eggs, facilitate fertilization, and support pregnancy. Key components include ovaries, produce eggs and hormones like estrogen and progesterone. Fallopian tubes, transport eggs from the ovaries to the uterus. Uterus, the organ where a fertilized egg implants and develops into a fetus. Vagina, the birth canal through which the baby is delivered and serves as the receptacle for sperm during intercourse. Hormones such as estrogen and progesterone regulate the menstrual cycle, ovulation, and the maintenance of pregnancy. Developmental stages. Gametogenesis, the formation of haploid gametes, sperm and eggs, through meiosis is essential for sexual reproduction. Spermatogenesis in males produces sperm, while oogenesis in females produces eggs. Both processes ensure genetic diversity and proper chromosomal distribution during fertilization. Fertilization. Fertilization occurs when a sperm successfully penetrates an egg, resulting in the formation of a zygote. This single-cell entity undergoes cleavage and develops into a blastocyst, which then implants into the uterine wall, marking the beginning of pregnancy. Embryonic development. The embryonic period, spanning the first 8 weeks, is characterized by the formation of primary germ layers, ectoderm, mesoderm, and endoderm, and the development of major organs and structures. This stage sets the foundation for the complex organization of the body. Fetal development. From the 9th week until birth, the fetus undergoes significant growth and maturation. During this stage, organs continue to develop and function, sensory and motor skills emerge, and the fetus prepares for life outside the womb. Hormonal regulation. Male hormones. Testosterone regulates sperm production and secondary sexual characteristics. LH and FSH, produced by the pituitary gland, control the production of testosterone and the process of spermatogenesis. Female hormones. Estrogen and progesterone regulate the menstrual cycle, ovulation, and pregnancy. Estrogen promotes the growth of ovarian follicles and prepares the uterine lining for implantation. Progesterone, produced by the corpus luteum, maintains the uterine lining during early pregnancy. Human chorionic gonadotropin, HCG, supports pregnancy by sustaining the corpus luteum and ensuring continued production of essential hormones. Menstrual cycle. The menstrual cycle is regulated by a complex interplay of hormones that control follicular development, ovulation, and the preparation of the uterine lining for potential implantation. The cycle is divided into phases, including the follicular phase, ovulation, and the luteal phase, each marked by specific hormonal changes. Pregnancy and birth. Hormonal changes during pregnancy support fetal development and prepare the body for labor. Hormones such as oxytocin stimulate uterine contractions during childbirth, while the placenta produces hormones that maintain pregnancy and support fetal growth. Conclusion. Understanding the key concepts of human reproduction and development provides a comprehensive view of how life begins and evolves from conception through birth and early postnatal growth. The integration of reproductive anatomy, developmental stages, and hormonal regulation highlights the complexity and precision of these biological processes. This knowledge is essential for advancing reproductive health, prenatal care, and overall understanding of human development. The subsequent chapters will build upon these concepts, exploring related topics such as genetics, the immune system, and the impact of lifestyle choices on health. The role of genetics in reproduction and development. Genetics plays a crucial role in human reproduction and development, influencing everything from the formation of gametes to the growth of an individual. Understanding the principles of genetics helps to explain how traits are inherited, how genetic variations arise, and how these variations can impact health and development. Genetic basis of inheritance. Chromosomes and genes. Humans have 23 pairs of chromosomes, which contain genes, the basic units of heredity. Each gene consists of DNA sequences that encode instructions for producing proteins. One set of chromosomes is inherited from each parent, providing a genetic blueprint for the offspring. Alleles. Genes exist in different forms called alleles, which can be dominant or recessive. Dominant alleles mask the expression of recessive alleles. The combination of alleles an individual inherits determines their genetic traits. For example, a person with one dominant allele for a trait, e.g., brown eyes, will express that trait, even if they have a recessive allele for another trait, e.g., blue eyes. Genotype and phenotype. The genotype refers to the genetic makeup of an individual, while the phenotype is the observable expression of the genotype. For instance, an individual's genotype for eye color might include alleles for both brown and blue eyes, but the phenotype will show the brown eye color if the brown allele is dominant. Genetic variation and mutation. Genetic variation. Genetic variation arises from mutations, recombination, and other genetic processes. Recombination occurs during meiosis, when chromosomes exchange genetic material, creating new allele combinations. These variations contribute to the diversity of traits in populations and are essential for evolution. Mutations. Mutations are changes in the DNA sequence that can occur spontaneously or due to environmental factors. Some mutations are neutral, some are beneficial, and others can cause genetic disorders. For example, mutations in the BRCA1 or BRCA2 genes are associated with an increased risk of breast and ovarian cancer. Genetic disorders and screening. Inherited disorders. Some genetic disorders are inherited in a Mendelian manner, following patterns of dominant or recessive inheritance. For example, cystic fibrosis is a recessive disorder, meaning an individual must inherit 2 copies of the mutated gene, one from each parent, to express the condition. In contrast, Huntington's disease is a dominant disorder, where only 1 copy of the mutated gene is needed to develop the condition. Genetic screening. Genetic screening can identify individuals at risk for certain genetic disorders or detect carriers of genetic mutations. Prenatal screening, such as amniocentesis or chorionic villus sampling, CVS, can assess the risk of genetic conditions in a developing fetus. Newborn screening tests can detect genetic disorders early in life, allowing for early intervention and management. The role of genetics in development. Embryonic development. Genetic information directs the formation of tissues and organs during embryonic development. Genes regulate processes such as cell differentiation, organogenesis, and body patterning. Disruptions in these processes, due to genetic mutations, can lead to developmental disorders or congenital anomalies. Epigenetics. Epigenetic modifications influence gene expression without changing the DNA sequence itself. Factors such as environmental conditions, nutrition, and lifestyle can affect epigenetic changes, which can be passed on to future generations. Epigenetics plays a role in regulating developmental processes and adapting to environmental changes. Genetic counseling and ethical considerations. Genetic counseling. Genetic counseling provides individuals and families with information about genetic conditions, inheritance patterns, and risks. Counselors help interpret genetic test results, discuss potential implications, and support decision-making related to reproductive choices and health management. Ethical considerations. Advances in genetic technologies, such as gene editing and genome sequencing, raise ethical questions about privacy, consent, and the potential for genetic discrimination. It is important to address these ethical issues thoughtfully to ensure responsible use of genetic information and technologies. Conclusion. Genetics is fundamental to understanding human reproduction and development. The principles of inheritance, genetic variation, and the role of genetics in development provide insights into how traits are passed from generation to generation and how genetic factors influence health and development. By exploring genetic disorders, screening methods, and ethical considerations, we gain a comprehensive view of the impact of genetics on human life and the future directions of genetic research and technology. The interplay between lifestyle choices and reproductive health. Lifestyle choices significantly influence reproductive health and overall well-being, impacting fertility, pregnancy outcomes, and long-term health for both parents and their offspring. Understanding how daily habits and environmental factors interact with biological processes helps in promoting healthier reproductive outcomes and informed decision-making. Nutrition and reproductive health. Balanced diet. Adequate nutrition is crucial for maintaining reproductive health. A balanced diet rich in essential nutrients supports hormone production, egg and sperm quality, and overall reproductive function. Key nutrients include folic acid, iron, calcium, and omega-3 fatty acids, which play roles in cellular function, hormone regulation, and fetal development. Folic acid. Folic acid is especially important for women planning to conceive. It helps prevent neural tube defects in the developing fetus, such as spina bifida. It is recommended that women take folic acid supplements before conception and during the first trimester of pregnancy. Weight management. Both underweight and overweight conditions can affect fertility. Excess body fat can lead to hormonal imbalances, impacting ovulation and sperm quality. Conversely, underweight can disrupt menstrual cycles and affect the production of reproductive hormones. Maintaining a healthy weight supports optimal reproductive health. Exercise and reproductive health. Regular physical activity. Regular exercise contributes to overall health and can positively influence reproductive function. Moderate physical activity helps regulate hormones, improve circulation, and manage stress, which can enhance fertility and support a healthy pregnancy. Excessive exercise. While moderate exercise is beneficial, excessive physical activity, especially in athletes or individuals with high training regimens, can negatively impact reproductive health. It can lead to menstrual irregularities in women and decrease sperm production in men. Substance use and reproductive health. Alcohol consumption. Excessive alcohol consumption can impair fertility in both men and women. In women, it can affect ovulation and increase the risk of miscarriage. In men, alcohol can reduce sperm count and motility. Limiting alcohol intake is advisable for those planning to conceive. Smoking. Smoking has detrimental effects on reproductive health. It can reduce fertility in both sexes by affecting hormone levels, damaging gametes, and impairing reproductive organs. Smoking during pregnancy increases the risk of preterm birth, low birth weight, and developmental issues in the baby. Drug use. The use of recreational drugs can interfere with reproductive health and pregnancy outcomes. Drugs such as cocaine and heroin can disrupt hormone balance, impair fertility, and lead to complications during pregnancy. It is essential to avoid illicit drug use and seek support for substance abuse issues. Environmental factors. Exposure to toxins. Environmental toxins, such as pesticides, heavy metals, and industrial chemicals, can affect reproductive health. These substances can disrupt endocrine function, impair fertility, and contribute to developmental issues. Reducing exposure to harmful chemicals and adopting safer practices can mitigate these risks. Workplace hazards. Certain occupations may expose individuals to reproductive hazards, such as chemicals or radiation. Occupational health and safety measures should be observed to minimize potential risks to reproductive health. Employers and employees should be aware of and address any reproductive health concerns related to the work environment. Stress and mental health. Impact of stress. Chronic stress can negatively affect reproductive health by disrupting hormone levels, impacting ovulation, and reducing fertility. Stress can also contribute to unhealthy behaviors, such as poor diet and substance use, which further impact reproductive health. Mental health. Mental health is closely linked to reproductive well-being. Conditions such as depression and anxiety can influence hormone levels and reproductive function. Seeking mental health support and managing stress through relaxation techniques, counseling, and healthy coping strategies can improve reproductive outcomes. Preventive health measures. Regular health checkups. Routine medical checkups, including reproductive health screenings and preventive care, are important for maintaining reproductive health. These checkups can help identify and address any issues early, support healthy lifestyle choices, and provide guidance on optimizing reproductive health. Preconception counseling. Preconception counseling helps individuals and couples assess their health, review lifestyle factors, and make informed decisions to improve reproductive outcomes. It includes evaluating nutrition, managing chronic conditions, and addressing any potential risks. Conclusion. Lifestyle choices play a critical role in reproductive health, influencing fertility, pregnancy outcomes, and long-term well-being. By adopting healthy habits such as balanced nutrition, regular exercise, and avoiding harmful substances, individuals can enhance their reproductive health and support successful conception and pregnancy. Understanding the interplay between lifestyle and reproductive health empowers individuals to make informed decisions and prioritize their overall health and well-being.